Okay guys, so today we're going to do our cell membrane lab. So I'm going to show you what you would have normally done in the lab, and I will give you your data, and based on that data you guys are going to do your analysis. So a couple points that you guys probably have reviewed on your background, but I'm still going to review with you guys, is when we're looking at the concept of diffusion, the size of the molecule or the molecular weight of the molecule has a significant effect on how fast a molecule can diffuse. Basically, the smaller the molecular weight or the lighter the molecules are, the faster they can diffuse, and the heavier the molecules are, the slower they travel. So to test this, we're going to be using two dyes. One is our methylene blue, and the other one is potassium permanganate. So what you would do, first of all, you will go ahead and search these two dyes, again methylene blue and potassium permanganate, and look for their molecular weight. So when you find that information, you guys are going to enter it on the chart in your lab, where it asks you for the molecular weight of these two samples. Now what you would normally do is you will grab a plate. So this plate contains agar gel in it. And if I turn it around, and I don't know if you guys can see that or not, but there's two little holes inside this gel. Now what we're going to do is we're going to add a small drop of methylene blue to one of these openings, and then potassium permanganate to the other one. Now what our goal is what we expect to see, or what our prediction is whichever these molecules are lighter or have a smaller molecular weight, they will travel a longer distance compared to the heavier molecule. So we're going to set up a small drop of each of these samples to each of the openings, and then let this sample sit for about 45 minutes. So I'm going to just start with my potassium permanganate, and you guys don't really need to label anywhere because the color is very distinct. So I'm going to add a small drop of my potassium permanganate to one, and again I'm going to add a drop of methylene blue to one, and then the potassium permanganate to my other openings, and then just let the sample sit, and let's see how fast these samples are going to diffuse. So again, 45 minutes for this sample. Now while we're waiting for this, we're going to do our next experiment, which is normally would have been an instructor demo. So for this one, we're looking at the sizes of molecules and see how the size affect the travel of molecules through the plasma membrane. So in this case, what we're using is an artificial cell. So these are known as the dialysis bag. Normally what they would look like, I'll show you a piece of this. It kind of look like plastic bags, tiny plastic bags, and you can open them up. We usually keep these dialysis bags basically hydrated, and we can use them to see if molecules can pass through them as they act as our artificial cell membrane. So here in this bag, what I've done is I have tied one end of this using a string. I want to make sure nothing leaks out. And then what I have added into this baggie is a solution of 10% starch. So here is my 10% starch. You can see the cloudiness of the solution in the container as well as in here. And then I went ahead and tied the other end of this sample as well. So before I do anything, what I want to do is I'm going to weight this baggie on my on my balance. And when I measure this, the weight of this baggie is 21.5. So I've already done that. So the weight is recorded as 21.5 grams. So the other part that you can see that you need to record is the color of this bag. And again, you can see the color inside this baggie is cloudy because of the amount of starch you have in the sample. Now the last part of this table that you have is asking you for the color of the beaker solution. So inside this beaker, what I'm going to add is a few drops of Lugol iodine. Now Lugol iodine is an indicator for a starch. So again, Lugol iodine, if you look at the normal color, and I can show you when I add it to the water. So if I add the few drops of Lugol iodine, you can see the color is yellow, right? So it's changing our water to yellow. Now, if a starch comes, not starch, if your Lugol iodine comes into contact with a starch, doesn't matter what condition it is, then your color of the starch will change to bluish black. Because Lugol iodine, the yellow solution, is an indicator for starch. And through this interaction, the color of the starch will change to bluish black. Or I should say the color of the indicator changes to bluish black. So what our experiment is going to do is we're going to take this baggie and we're going to place this baggie inside our Lugol water, Lugol iodine water, and observe the final result. So let's look at it in terms of prediction. So if you're looking at the starch baggie, right, and this starch bag gain weights, that weight is usually indication of the water molecule diffusing or using osmosis to get into my baggie, therefore making my baggie more heavy. The other option is my baggie weight goes down after 45 minutes. In such a scenario, that implies that the water has moved out of my little baggie into my solution and as a result caused the weight loss. What if the solution inside my baggie changed? So right now, again, if you recall, it was a cloudiness that is associated with the starch solution. So let's say the color of my baggie changed to the bluish black. In that scenario, it means iodine successfully passed through the cell membrane and was able to interact with my starch. If the color of the starch stays the same, that means the iodine did not pass through the membrane and therefore no color change was observed. Our last scenario prediction, or I guess the two options would have, is if the solution of the beaker, which is currently yellow, stays yellow, that means a starch was not able to get out of your baggie and get into the solution. And if the beaker solution, which is currently again yellow, changed to bluish black, that means that the starch molecule was able to get out, and considering that observed color change, it means that the starch interacted with our indicator iodine and observed the color change. So basically what I want to see again, the goal of this experiment is to see which of these molecules that are involved, my water, the starch, and my iodine, are small enough to either diffuse or move into my cell or out of the cell based on their concentration. So we're gonna let this beaker sit also for about 45 minutes. So we're gonna set this aside. The next experiment we're gonna do is using the concept of osmotic pressure. So again, if you are unfamiliar with that, please review the background information I have provided for you. But the basic idea behind osmotic pressure is basically the pull your solute molecule have on your water. The more solutes you have in a location, the more water wants to move in that direction. Osmotic pressure is extremely important for our body. It is what causes a lot of physiological changes in our body when there is fluctuation in our iron concentration, when there is a significant fluctuation in our salt concentration. So it is extremely important for our body to maintain a very specific solute concentration intracellularly and extracellularly to make sure that water does not significantly shift from one place to another. Now, keeping the whole osmotic pressure in mind, what we're doing basically is we're looking at 3 samples and observing which of these will have the highest osmotic pressure. Again, osmotic pressure is pull solute molecules have on water. So right here for you, I have 3 samples of sucrose solution. We have a 10% sucrose solution, we have a 20% sucrose solution, and we have a 30% sucrose solution. So basically that means this has 10% of sugar, 20% and 30% sugar. Now take a moment and think about it in terms of prediction. Which one of these do you think will have the highest osmotic pressure based on their concentration? Now for us to figure out what is our osmotic pressure and answer the question, what I have done partially is we have created these little baggies. So this baggie that I have is a 10% sucrose solution. I have another one which is a 20% sucrose solution. And I'm going to show you how to actually make the baggies, since I haven't shown you. I'll show you in a second how to make the baggies with our 30% sucrose solution. Now the idea here is, after I'm done setting this up, I'm going to place each of these little baggies inside my, inside a water solution. This is just simply DI solution or DI water. So I have the 10%, 20, and 30%. After weighing my little baggies, I'm going to drop them into the water and wait for 45 minutes. Again, why am I doing this? So the goal is, this is pure water and this is sugar, right? So regardless of, am I talking about 10%, 20%, and 30%, water wants to diffuse into my baggies, and therefore we will observe a gain in weight for all three baggies. The question is, which one of these baggies do you expect to see having the highest weight gain versus which one do you expect to have the lowest weight gain? So take a moment, think about it, make your prediction in that term. Now again, what I'm going to do, I'm just going to take a second and show you how to properly set up these tubes. So here is a piece of dialysis baggie. I'll take a couple pieces of strings. And all you do is kind of like wrapping a piece of candy. So you're going to take the your little baggie and you're going to twist, 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 and you're going to wrap it. You're going to tie a knot on it. And again, this is really an important step, because if you don't secure this properly, then it would leak out and your experiment would be incorrect at the end. So I'm going to open up my little baggie, mix my sucrose solution, and then I'm going to add the fluid. You don't want to bring it all the way to the top, because if you do that, then you don't really have the option of adding any water as the experiment starts. So see, I have about an inch empty on the top. So then I'm going to twist again, twist, twist, and I'm going to close the other side as well. It's literally like closing a candy, or piece of candy, it's not a candy. Every time I do that, it reminds me of I Love Lucy. If you guys have no idea what I'm talking about, take a moment and do a Google search on I Love Lucy candy factory. Okay, so now I have my three baggies. I have a 10% baggie, a 20% baggie, and a 30% baggie. So I've already rated the other two for you. So the first one is a 15.5 gram weight. My second baggie is 12.6 gram in weight. And the third one is 13.5 gram in weight. So again, I'm gonna at the same time, or simultaneously, introduce these baggies into the water and start a timer that is basically run for 45 minutes. I'm gonna weight these baggies again, and I'm going to see which of these baggies have gained the least weight versus the most weight. And based on that, I can answer my prediction how osmotic pressure or how percentage of sugar affect my osmotic pressure. Okay guys, so let's go ahead and take a look at our results for our three experiments. So the first experiment we did earlier was on how the molecular weight affects the diffusion rate. So right here we have our two samples. So we have the blue one, which is our methylene blue, and the red ones is our potassium permanganate. So what I'm going to do is I'm going to take a ruler and, using the centimeter aspect of this, measure the diameter that the stain has traveled. So for the first sample that I have, which is my methylene blue, I have 1.2 millimeters, or sorry, 1.2 centimeters in terms of travel. And in terms of my potassium permanganate, I have 2.5 centimeter in terms of transfer. So enter that data. Again, 1.2 centimeter for your methylene blue and 2.5 centimeter for our potassium permanganate. Now, based on that, answer the question you have. Does the prediction of lower weight implies faster diffusion rate for in your lab activity? Okay, the next one that we did was our experiment to see how size affect in terms of diffusion passing through the plasma membrane. So right here, again, we had the original sample. We had a baggie of the starch, which has the weight of 21.5 grams. The original color was cloudy, and the water of the beaker was yellow. So right here, I have completed the experiment. So here is our baggie. So you can see that our baggie went from that cloudiness to bluish black color. So in terms of the color of the starch bag, for the final you can write black or bluish black. I have taken a moment to weigh this bag before, and the current weight is 23.2 grams. So 23.2 grams, which means the weight has been increased. And then lastly, you can see the color of the beaker remains as yellow. So there is no color change in terms of the beaker. So when you guys answering these questions regarding this experiment, do keep in mind our goal is to identify if each of the three molecules involved moved. What is the means of movement? Is it diffusion or osmosis? And if there was a diffusion, what is the main reason that diffusion was present or absent? Again, always think about in this experiment in terms of size. Now, the last experiment we did was our three baggies of three different types of sucrose solution. I got kind of water over here. So we have the 10%, the 20%, and the 30%. So this experiment also ran for about 45 minutes, and I did take a moment to weight these bags to see what the final weight for each baggie is. So the first baggie, which is our 10%, the current weight is 15.9 grams. Our second baggie is 13.6 grams. And our third baggie, which is our 30%, is 15 grams. Now you guys are going to do your calculation for the percentage change. That information is found in your activity, so take a moment, look at it, and then finish the lab by doing a bar graph. I expect to see on your bar graph 3 bars, one for the 10%, one for the 20%, and one for the 30%. This is your x-axis, and the y-axis of your graph should represent the percentage change. You are, again, only expected to see 3 bars. I do not want to see dots or line graphs. None of those are acceptable for this experiment. The data should not be connected, and the data should be represented in percentage change, not initial and final weight. Though those information were needed to collect your data and to basically do your calculation, but the graphing only requires you to focus on the percentage change. And then after you've done that, complete the questionnaires associated with this lab activity. So that's about it in terms of our 3 main lab activities. I do want you guys to also think back about the whole concept of hypotonic, hypertonic, and isotonic. These were the aspects of the information that were associated with how your cell will be affected if the solution around your cell will be changed. So there are questions in that lab activity that you are required to complete based on your understanding that your normal cell is present in a solution of 0.89% NaCl. Some people round it up to 0.9% NaCl. That is a typical isotonic solution, which means that is the environment your cells are going to be happy and will survive and be basically keeping their normal shape. But when we're comparing a hypotonic or hypertonic, the movement or shifting of the water in or out of the cell causes a significant change in the cell shape, causing it either to shrink or crenate, or for the cells to swell up and burst as a result. Based on your understanding of what is identified as isotonic solution for our cells, I do expect you guys to make prediction or have a good understanding of which of the given solutions in your lab activity are isotonic, hypotonic, and hypertonic, and how each of these solutions are going to affect your cell. So that's it for me. If you guys do have any questions, please contact me.